Thank you for the invite. I feel like I should jog up and with that music. I'm very jealous of the speaker there. I want him to do my voicemail. I'm Marc Odrich. I'm the Chief Medical Officer for LENZ Therapeutics. I'm going to talk today about our particular miotic. I want to point out that we are basing our miotic on the science of what is the optimal pupil diameter. And as I think I've mentioned to many different audiences, the Sharman and Whitefoot data clearly suggests a sub 2 millimeter pupil is ideal if you expect to get a significant increase in the depth of focus. This has been clinically demonstrated on the right side of this slide by showing you not just theoretical but actual patient data looking at specific eyes, looking at different pupil diameters, and demonstrating very good correlation with the ideal. The ideal presbyopic drop is pupil selective. And by pupil selective, I am specifically saying avoiding the ciliary muscle. And that's one of the key features of our active aceclidine. Aceclidine was not brought into this country because in 1972, Steve Drance correctly identified the fact that it didn't do very much for the scleral spur and it didn't do very much for the trabecular meshwork because it avoids the ciliary body and the ciliary muscle. Those two characteristics, iris sphincter targeted and avoidance of the ciliary muscle, account for the very benefits that make it an ideal miotic and at the same time make it a lousy glaucoma drug. And so it was never brought into the US. However, there are decades of experience with aceclidine in Europe, and its safety profile is well known. When you look at aceclidine, one of the interesting things to support this is that the EC50 ratio, which is what you're seeing here, is the selectivity to the iris sphincter is profoundly in favor of the aceclidine versus the carbachol and pilocarpine-based medications. So this is a profound difference from both carbachol and pilocarpine. In addition, to prove that point and make sure I'm just not whistling Dixie, what you have up here is the change in myopia. And what you're seeing is with a 2% aceclidine, you have an almost undefinable myopic shift, meaning the lens iris diaphragm is not pushed forward and you don't have a ciliary muscle effect, so you would not expect aceclidine to cause any change in distance vision, and in fact it doesn't in our clinical trials. Additionally, you can see there's between a 1 and 1.5 diopters of motion causing myopic shift with the other two drugs at these particular concentrations, which is what was studied. This is from the peer review. This was not done by us. Biomarkers. So vision is great, and I'll show you our vision data and our phase 2C data, but you can't fake pupil size. And what this demonstrates is that for 10 hours, we are sub 2 millimeters, or just 2 millimeters. That means that you have a statistically very significant effect. The other thing you'll notice is that we have two formulations, one which includes a pinch of brimonidine. I said that intentionally. And what you find is that, in fact, you have a small prolongation of the miotic effect by adding a little bit of brimonidine. So how does it work? Well, it works really well. 3-line improvement with no lines of 1 line or less of vision loss. And what you can see is that we're talking in the in the 70s and the high 60s for the two formulations up to 5 hours. And then the interesting thing is the aceclidine alone tends to drop slightly while you maintain or even improve a little bit with the combination product. Both of these are class leading. You'll also notice our low placebo response rate, which is indicative of a well-controlled study. This was a crossover design, so we're looking at the same patients over and over again through different testing. It is extended duration, out statistically separating at 9 hours, and then 94% of the subjects are achieving 20/40 or better. Finally, when you start to look at a comparison, and the data that we have currently is the approved Vuity data, and this is taken from the package insert, what you see is the efficacy comparison in a placebo-controlled studies. You see the difference between these drugs. We're talking a multiple in terms of effectivity, and we're talking out to 10 hours, highly significant responder rates. So we are in phase 3 currently, and I'm looking forward to bringing this as a best-in-class potential as a pupil-selective miotic, all eyes, all day long. Thank you.